So welcome back to our next lecture. The last time we met, we were looking at nucleoside analog. So I'll give 3 more nucleoside analog and I'll be done here. So I'll give an example of a nucleoside. Okay, let's say we pick a cytosine based nucleoside. Okay, so this is our cytosine attached to ribose sugar. This is ribose sugar. Okay, CH2 here, hydroxide there. So this is it. What is this? We would say, what would we say? We would say this is the site D. Site D. This is site D. So we can make adjustments to this site D. Okay, we can make adjustments to this site D. Okay, so one of the adjustments that we might want to do is to remove the hydroxide here. Remove the hydroxide here so that you only have the hydrogen. But the same hydrogen that you have removed, or let's remove the hydroxide and the hydroxide so that we remain with the hydrogen only. So the analog that results. Okay. So all that we have changed, is this. It's on the sugar. On the sugar. So we have removed the hydroxide, so we have remained with the hydrogen there. The hydrogen there. The rest remains the same. Okay, so what we have produced, we would say at 2' and also at 3'. Remember, 1' is here, 2' is here, 3' is here, 4' is here, 5' is there. So at 2' we have removed the oxygen. At 3' we have removed the oxygen. Okay, so we'd say 2 deoxys if you want. So 2 deoxy. Okay, that's a prefix. The parent is cytidine. Cytidine. Okay. So sometimes people would want to make this the shorthand to say 1D and another D and a C, DDC. Okay, DDC. Okay. So the common name that is used is Sulfur-based. Another analog. Okay, another analog. Okay, that's the other analog that can arise from the same. Okay, the other analog that can arise from the same. We might decide. Okay, we might decide to remove these hydrogens. Okay, we have removed these hydroxide, but upon removing the hydroxides, let's remove the hydrogens also. So if we remove the hydrogens, for this to have a valence of 4, carbon to have a valence of 4, you must have a double bond pair. So in that case, what you produce, so you see that we are just playing in the lab. Okay, in the lab we are just playing with the structure, trying to create analogs, trying to create structures that are similar to our, where is my So we are just trying to create structures that are similar to our nucleosides. So this is our nucleoside, CH2, and hydroxide. So we've created a double bond. Okay. After creating a double bond, we further create, we put a phosphorus here. Okay. We put a phosphorus. So, in other words, we have created at 5 position, there is fluoro. Okay, fluoro. Okay. But at 2' and at 3', we removed the hydrogen. So we would say 2 removal of hydrogen. So dihydro. No, it is dihydro. Dihydro, that's the removal of hydrogen, but there are 2. So dihydro. Okay, but remember we initially removed the hydroxide. So 2', 3', again 2 dideoxy. Okay, the whole compound is a cytidine, dideoxy cytidine. So that's another analog of cytidine. Okay, sometimes this is called the elutabine. Okay, that's another drug. Okay, this salcitabine is a drug. Elutabine is a drug that is used to treat HIV. Okay, HIV. Although some of these are being phased out because of their toxicity issues. Okay, because of their toxicity issues. Okay. Then the last 2 that we want to give, maybe last 2 is that the same cytidine, we might decide, okay, to remove the oxygen here. We remove the oxygen, replace it with carbon, and remove the oxygens here. Remove the oxygens at these 2 positions. This is it. 1', 2', 3', 4', 5'. So if you do that, okay, so I have removed oxygen here, I've put a carbon. So I'll just remember this is a carbon. Okay. Then I have CH. No, I'm sorry. Okay. The oxygen is still there, but instead I remove the oxygen here, and I remove the carbon here. There's a carbon here. I remove the carbon, I replace it with sulfur. Okay, I replace it with the sulfur. In that case, what have I done? In that case, this is 3'. At 3', I've put a sulfur. Sulfur is said to be thia. Okay, the prefix for sulfur is thia. Okay, then at 2' and 3', I have removed the hydrogen. Sorry, I've removed the oxygen. So 2 removal of oxygen. So 2 dideoxy. Okay, dideoxy, and then the parent is cytidine. Okay, parent is cytidine. Okay, so if you want, you can say, if we want to write the shorthand, okay, you can say 3T and the C. Okay, 3TC. Some people call this as lamivudine. Lamivudine, which is a drug that is used to treat, again, HIV. Okay. Lastly, we can also modify the same lamivudine. We don't change much, we just introduce it. The organic chemists will tell you that all these modifications were done to improve the drugs, okay? To improve either the efficacy or reduce the toxicity levels, the toxicity that they were causing. Okay? But that's for organic chemists, not for biochemists. Okay, so here we have sulfur, okay? Here we have CH2 dioxide. So, and then we have introduced fluorine there. Okay, so we can say this is 5-fluoro, 5-fluoro, okay, then 5-fluoro, then 3' thia, okay, then 2', 3' dideoxy cytidine. Dideoxy cytidine. Okay. Others call it as F for fluoro. Okay. Then T for thia, and C for cytidine, FTC. Okay, but others call this as Citabine. Okay, all these are but just drug analogs. Okay, drug analogs. Okay. Right, we have just looked at the pyrimidine-based nucleoside analogs. What about purine-based? What about purine-based? Can we just look at two purine-based? Two purine-based. Let's look at adenine. Let's look at adenine. Okay. Adenine is like this. Wait a minute. Adenine is not like that. If I start like that, I'll run out of space. So now I start like this. Out of space. Okay. So adenine. So we can create a drug from here. Okay, we can create a drug from here. Well, let me start with guanosine. Okay, this is our guanosine. Okay, right. If we look at guanosine. Okay, guanosine. So what we can do to the guanosine is to remove this oxygen. Okay, if we do that, okay. Right. If we do that, so we have carbon there. The terminal CH2 hydroxide. Okay. And then remove the hydroxide, remove the oxygens. Then the resultant oxygens, remove the resultant hydrogens, remove them. So then it means we have to create a double bond here. Okay. Okay. So we have removed the oxygen here, replaced it with carbon. The oxygen and oxygen are removed, resulting into hydrogen. The hydrogen are also removed. This is called the carbovia. Kabova, no, Kabovia, okay, Kabovia, okay. Right, but this can also be modified if you want. You can introduce a cyclopropylamine here. You can introduce cyclopropylamine here. If you do, this is what you create. Okay, so remember an amine reacting with the ketone, the oxo group is going to be replaced. It's going to be replaced. That is a cycloamine introduced there. Okay. Then, I'm sorry, Kabovia, I've written it wrongly. Please correct me. Remember where we're coming from, it has to be at position number 9. Okay? It has to be at position number 9. I put it at the wrong place. Okay, sorry for that. It has to be at the position number 9. Sorry for that. Sorry for that. Okay, it has to be at position number 9. The sugar must be attached to position number 9. Okay, similarly here, it has to be attached at position number 9. Sorry for that. Okay, what we have created is a bacavia. Another ARV drug. Okay, right. So these are nucleoside analogues. These are nucleoside analogues. Right, before we proceed, let's look at briefly at the features of purines and pyrimidines. Okay, before we go to nucleotides, let's look at the features of nitrogenous bases. Nitrogenous bases. What are the features? Okay, nitrogenous bases. One, we see that they are hydrophobic. Okay, they are hydrophobic. Both the purines and pyrimidines are hydrophobic. Okay, they are water hating. Okay, you can see there are a lot of carbons in the structure, so they are hydrophobic. Secondly, what you also see is that the pyrimidines are planar. Pyrimidines are planar, while the purines are almost planar. They are not fully planar, but almost planar. Okay. All right. Thirdly, the other feature that you see is that both the purines and pyrimidines can undergo the process that is known as tautomerization. Formation of tautomers. They can form tautomers. Okay, the organic chemists must have done a good job on the tautomerization, formation of tautomers. Okay, for instance, let's look at cytosine. Okay, when you look at cytosine, this is our cytosine. Okay, that is our cytosine. Okay, the hydrogen one pair there. Okay, we can say that this hydrogen, let me write the hydrogens this way so that we see that they are isolated. This hydrogen, remember there's a lone pair here. The lone pair can abstract this hydrogen so that the hydrogen is attached there. Okay. If that happens, it means that this double bond cannot be there. if this abstract a proton, then this double bond has to move to that position. Okay? Right, what I'm saying is that this can abstract a proton like that, and if that happens, this has to move like that. So in the process, you do not have two hydrogens, you only have one hydrogen, and a double bond is created there. Okay? The double bond there is lost, and the nitrogen there. It's the same cytosine. It's the same cytosine, except that now it's in a different form. Okay, it's in a different structure. So that right. Okay. So with reference to this group, this is cytosine in form of a ketone. Sorry, a keto form. It's a keto form of cytosine. Okay. I'm sorry. It's a keto form. But in terms of minor group, this is a minor group. It's an amino form. I'm sorry for that. How can I say that? This is a minor form. But in terms of what has been formed, this is an imino form. Okay? It's still cytosine, but in the imino form. So, cytosine can exist as an amino form or an imino form. Okay? An amino form or an imino form. This process is called the tautomerization. This is called the tautomerization. Okay? What predominates, okay, in nature, when you look at nucleic acids, what predominates is the amino form for cytosine. Are we together? Yes. So these are tautomers. The amino form, they are tautomers. Tautomers, we just sort of intramolecular movement of a hydrogen between one carbon. So there's only one carbon here. So this comes here. Okay? So the other examples that we can think of, let's look at thiamine. Okay? Let's look at thiamine. We've looked at cytosine. Let's look at thiamine, whether it can exist as a tautomer. So this will be our thiamine. Okay, that will be our thiamine. Okay. Thiamine as a methyl group there. So remember, tautomerization, the hydrogen must move just one carbon. Okay. So it's possible that, remember, this oxygen has a lone pair. This can come and abstract a proton there. Okay. If it abstracts a proton, then this means the double bond can't be here, because the oxygen will be positively charged. So this can come here. If that happens, then you have that scenario. Okay, the double bond has come here. Okay. Remember the lone pair, don't forget the lone pairs. Okay, so basically that's what has happened. So this is also tautomerization. Okay. So, with reference to this, this is sort of a ketone. So, this is thiamine, which is existing in form of a keto form. Okay. This is also thiamine. Remember, there is methyl group here. This is also thiamine, which is existing as an alcohol and a double bond. So, we would say that this is a keto form. Thiamine, which is a keto form. Which is the ino form. Ino form. Alcohol and a double bond. Alcohol and a double bond. Ino form. But we can also say that this is an amide. This is an amide. You can say it's a cyclic amide. A cyclic amide is called a lactam. Lactam. A cyclic ester is called a lactone. cyclic amide is called the lactam. Okay, the lactam. Okay, right. Okay, and this would be a lactine. Lactine. So thymine can exist as keto-ino or lactine. What predominates is a keto form for thymine or a lactam. What predominates is a keto form or a lactam. Okay, if it changes to a ino form, this can bring about mutation, as we shall see later. Okay, that's about it. What about what remains? Uracil the same. Okay, uracil can undergo keto-ino form. Similarly, if you look at adenine can have an amino and imino form. Okay, guanosine can have a keto-ino form. So all these undergo tautomerization. Remember, we're looking at the features of purines, pyrimidines, that they have or they are able to form tautomers. Okay, but also what we see is that both purines and pyrimidines, are both they're both hydrogen donors and hydrogen acceptors. This is very important. Hydrogen acceptors. Okay, I'll just give one example. The rest is the same. Okay, if you look at cytosine, I'll write cytosine here. This is it, my cytosine. Okay, my cytosine. Okay, this is my cytosine. Okay, there's a lone pair here. There's a lone pair here. There are two lone pairs here. The first topic we looked at was one of the features of water is that it's able to form hydrogen bonds. So if you check, this oxygen has these lone pairs. So these lone pairs can accept a hydrogen. These lone pairs can accept a hydrogen. These hydrogens can be donated. These hydrogens can be donated. Remember, they are attached to a highly electronegative element. So this is hydrogen donation. This is hydrogen accepting. Are we together? Hydrogen accepting, hydrogen donation. Okay. Similarly, if you look at uracil, okay, that is uracil. Remember, there are two lone pairs here. So you can say this can receive proton. This can receive a proton. This can donate a proton. So uracil is also a hydrogen donor, can donate